Now we'll be studying blood groups in detail. So he is the Karl Landsteiner. Okay, some write Karl and some write it as a Carl. Karl. Okay, so the Karl Landsteiner, he reported first time ABO blood groups in human beings, A, B and O blood groups that were discovered by Landsteiner, while AB blood group was found out by D. Castello and Stini. Okay, and ABO blood groups, they are determined by gene. There are three alleles, IA, IB and IO of the gene. Protein produced by IA and IB alleles, they are called as antigen A and B respectively. Okay, so it is of 5 types: type A, type B, type AB, type O, and RH positive and RH negative. Okay, as I have told you that it is based on the presence or absence of the two surface antigens that induce immune response on RBC, namely A and B, and similarly plasma contains two antibodies which is produced in response to antigen, namely anti-A and anti-B. Okay, see, blood group A, it has antigen on RBC A, but the antibody in plasma is anti-B. Okay, and it can donate blood to A and AB. That means a person with the blood group A can donate to the A and AB, but can receive blood from A and O. Okay, now come to B. B, it has B antigen on the RBC and the B and the anti-A antibodies in the plasma, and it can donate blood to B and AB, but can receive from B and O. Okay, blood group with AB, it has antigen A and B on RBC. Okay, but the antibodies in plasma are nil. It can donate blood to AB, but can receive from A, B, AB and O, and it is a, you can say that it is a universal acceptor. And then comes O. It has antigen on RBC, no antigen on RBC. It has antibodies anti-A and anti-B, so it can donate blood to A, B, AB and O, but it can receive blood from O only. Now, if you belong to a blood group A, you have A antigen on the surface of your red blood cells and the B antibody in your blood plasma. This is just the summary. Okay, and the blood group B, if you belong to the blood group B, you have B antigen on the surface of your red blood cells. As you can see in this diagram, this yellow part, it is a B antigen, but it has A antibodies in your blood plasma. This is your plasma and it has A antibody, just opposite. Okay, and what happens in the AB, if you belong to AB blood group, you have both A and B antigens on the surface of your red blood cells. See, this is B and this is A. Okay, but neither A nor B antibodies in your blood plasma. Now what happens in blood group O? If you belong to the blood group O, you have neither A nor B antigen on the surface of your red blood cells, but you have both A and B antibodies in your blood plasma. Always know that antigen A, it reacts with anti-A. Okay, and antigen B, it always reacts with anti-B, and the mixing of the blood with interactive antigen and antibody, it causes agglutination, okay, of RBC. O group, as I have told you that it is universal donor. It can donate to any blood group, any patient with a blood group of any type. Okay, and AB, it is a universal recipient. It can receive from any person. Okay, O group, they are called as universal donor as they can donate blood to person with any blood group, and AB group persons, they are called as universal recipient because they can accept blood from all groups. Okay, now comes RH positive and Rh negative. Rh means rhesus factor is an antigen that is found in RBC. Okay. A protein is named as rhesus antigen is present on the surface of the red blood corpuscles in many persons. It was discovered by Landsteiner and Wiener in the blood of rhesus monkey. Hence, its name is. Depending on the race, 85 to 99% of the white population have this rhesus antigen, and they are called as Rh positive. Others who do not have this factor, they are known as Rh negative. Rh positive is always dominant over Rh negative. Okay, please note that. And formation of Rh protein is controlled by a dominant gene, which we call it as a R. Thus, RR, that is capital R is a homozygous condition, and capital R, small r is a heterozygous condition. And this person are dominant and are Rh positive. And small r, that is homozygous, they are recessive and Rh negative. Okay. But you know what, both in Rh positive and Rh negative individual, they are phenotypically normal, but the problem arises during blood transfusion and pregnancy. Okay, that we'll see. See, this is incompatibility during blood transfusion. Okay. In this, what happens is the first blood transfusion of Rh positive blood to the person with the Rh negative blood causes no harms. As you can see, this Rh positive to the Rh negative, it causes no harm, because the Rh negative person, it develops anti-Rh factors or antibodies in her blood or in his blood. In the second blood transfusion of Rh positive blood to the Rh negative person, the latter's anti-Rh factor, they attack and destroy the red blood corpuscles of the donor. Okay, so Rh positive to Rh negative is not advisable, but Rh positive to Rh positive can be done. Okay. So I hope it is clear to you that how incompatibility takes place during blood transfusion. And then this is what I told you, that it occurs in pregnancy also. It can arise during pregnancy. If an Rh negative woman, it carries an Rh positive baby. Okay, if blood cells form from the baby travels across the placenta, because placenta is the connecting thread between women, mother or the baby. So if the blood cells from the baby travels across the placenta, the women's immune system will see Rh positive cells as a threat. And a specialized WBC will make antibody designed to kill Rh positive blood cells. If the woman later conceives another Rh positive, that means second Rh positive baby, her immune system will flood the fetus with antibodies. These antibodies then can destroy the baby red blood cells, and if left untreated, this can result in severe anemia or even death of the baby, because the immune system of the mother will treat baby as a foreign body. Okay, so this is called as hemolytic disease of the newborn. This is how Rh negative blood cells of a mother, and this is the Rh positive blood cells of baby. So this is known as the Erythoblastosis Foetalis. This is Rh incompatibility between Rh negative blood of the pregnant mother and the Rh positive blood of the fetus. And Rh antigens do not get mixed with the maternal blood in the first pregnancy, because placenta, it separates the two bloods. Okay, but in the second pregnancy it causes. Okay, but at the time of the first delivery, there is a possibility of the exposure of the maternal blood to the small amount of Rh positive blood from the fetus. This includes the formation of Rh antibodies in the maternal blood. and in case of her subsequent pregnancy, that is second or the third, Rh antibody from the mother can leak into the fetal blood and can destroy the fetal RBC. This is fatal to fetus or can cause severe anemia, and this condition is known as Erythoblastosis Foetalis. Okay. Erythoblastosis Foetalis can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of first child. So this ends with the second section of the circulatory system. So thank you and stay tuned and keep watching Edupedia Word videos.